it will be in July when the weather is lovely in Edmonton. It'll be 20 degrees, green, perfect. Okay. Also, don't be afraid to come in the winter. It's really not that bad. You just need a good coat. Okay, so today what I want to talk to you about is a problem that I would classify it as kind of a data science problem. I want to motivate it by some specific scientific problems. So these are problems that come from collaborations that I have with experimentalists. I'm not going to solve these problems for you. I just want to show you the motivation, okay? Because I think that it's really important to have a real scientific problem you're trying to solve when you're developing a method for something, right? Because we can easily fall into the trap of inventing a method that can solve a couple of toy special cases, but science is very unforgiving. So you're really forced to contend with the real problems and struggles when you have a real collaboration to guide your methods. So this is a methods talk, and this is a machine learning institute, right? So I hope that this topic is interesting to everybody. So the problem I'm trying to solve is tracking the position in microscopy videos. So this particular problem I'm about to show you, I also would like to track the lineage. What lineage of what? I'll describe that. But this, I'm nowhere near doing this. Okay, really, all I'm going to talk about is doing this. It's already very hard. Okay. So this is a project with Amy Gladfelter. She's no longer at UNC. She's now in Duke. She's a biologist at Duke, and this Grace McLaughlin is her graduate student. And so Grace is the one who took these lovely videos. We study a certain kind of fungus. So fungus has a life cycle. We might think of this as fungus, right? That goes into our soup. But part of the life cycle of fungus is the mycelium. It's this branch structure. So here's just something from Wikipedia. This is a big mycelium in coffee grounds, right? So in my kitchen, right, I didn't throw away the coffee grounds. I left it out, right? That's what's growing on it. Just kidding. Okay, the mycelium that we're studying is much smaller, okay? Much smaller. This is hundreds of microns, okay? So there'd be this is like one coffee ground. I don't think that's like an exaggeration. The coffee ground, they're probably much bigger actually. Anyway, this is one cell, and what you're seeing here, these little dots, are nuclei. Okay, so what's our textbook understanding of a cell? Sphere, right? Little sphere inside the big sphere. Little sphere is the nucleus. What's special about the nucleus? Has the DNA, right? That's pretty special. That's pretty important. Why would one cell need hundreds of copies of DNA? It's a pretty weird cell. Fungus is weird, right? That's the whole thing with fungus. It's like this beautiful, and it's a combination of beautiful and weird. I really like studying. Okay.
A single cell has this amount of nuclei.
Yeah, that's right. And so we're only looking at half, so it's radially symmetric. Just extrapolate. So these nuclei are moving by molecular motor transport, and the cell tries to maintain a constant density of nuclei. But that's not precise, right? Because these are moving stochastically. These tips are growing. I will show you other videos where you can see the growth, but it's too short to see the growth here. And these nuclei are dividing, right? So this is a big self-organization problem. How do you maintain this constant density of nuclei in this sort of constraint? So what we want to do is track these nuclei through time, and we also want to track when they divide and who the mother and daughters are, right? Tracking the mother and daughters that were divided, that's lineage. That's hard. we're not quite there. Right now, all we want to do is track the motion. But they are in this quasi one-dimensional domain, and they cross over each other. When they cross over each other, who goes where? We don't know. Well, these nuclei are identical. Visually, there's very little difference. Okay, so that's what we want to be able to do. Here's another video. As I promised, we'll show you the. This is fast, right? So now we can see it growing. So we also have to track these in a growing domain. We have to estimate this domain in order to successfully track, it turns out. So when two things cross, because they undergo molecular motor transport, usually they have a velocity. And then when they uncross, we can use the velocity to figure out who goes where. Our eyes do this naturally, automatically, right? If you have, I don't know, two frisbees that cross each other, two birds that cross, right? When they uncross, you know which one goes where because they keep going with the same velocity. But to use velocity to track, how do you estimate velocity? You need at least two successive time points, right? In order to get velocity. So it's this loop. You need both, right? Okay. I see some nodding, so I've at least convinced some of you that that's an interesting problem. Okay, so here's another problem. This is with a biologist, Greg Jed, at the National University of Singapore, and my former postdoc, Yonatan Ashanafi. How many of you have heard of diatoms before? I'm not surprised. A lot of people, how about diatomaceous earth? You use it in your garden probably sometimes. It kills something, some kind of pest. I can't remember. Anyway, diatoms are single-cell organisms. These are much smaller than the whole fungus cell, but much bigger. So this is what, like 20 microns? So they're much bigger than the nuclei I showed you in the previous slide. This is agar. So it's just like a little petri dish. We don't understand how they move. That's kind of our scientific question. We understand the mechanism of how they move. There's not many organisms that we have not resolved locomotion for a single-celled organism like cilia or flagella. There's all these mechanisms that they use. It has something to do with excreting polymer hydrogel, this goo. They like excrete goo, and somehow that makes them move. We don't really know. They're really cool cells. Okay, so what's interesting about diatoms? They produce something like 50% of the Earth's oxygen, so we really need them. They exist in coastal environments, mostly at the poles. Some of them are photosynthetic, and they exist in this really interesting transition zone where they move from being photosynthetic to moving. Why would they want to move? Well, they're going to search for food, right? That's the most basic reason why any organism would want to move, right? So these are no longer photosynthetic. They're also very beautiful. Diatoms have they're like little crystal submarines. They have this beautiful crystalline structure. Their cell walls are the silicate, almost like a glass, right? Glass is silicate, right? I don't know these words. And so one of the things to note here is that they'll often spontaneously switch direction, like that one right there just did. And so they go at like a relatively constant velocity. They might turn a little bit, and then sometimes they reverse direction. And then they collide a lot, and sometimes they're moving together, right? So they're difficult to track. But if we're going to explore mechanisms for how they move, our reason to track their motion is to get statistics about like how often do they reverse? What speed do they go? Do they go faster when they move together or slower, right? When they collide, what happens? That kind of thing. So that's why we want to track their motion. So again, we want to be able to use velocity to resolve what happens crossings and uncrossings. We also have objects that enter and leave the field of view. Our methods for identifying where objects are not perfect. I'll talk about that in a little bit. So we'll have a lot of missing observations, and we need to be able to deal with that. In both of those cases that I showed you, the velocities are switching, right? Like the diatom's reverse direction. That complicates things because basically we're going to have hidden states or latent states in our motion model, and we need to be able to resolve those. And here's a couple of popular ways of resolving that kind of thing: expectation maximization or switch point. So we need a framework that can use those things pretty naturally. Okay, so I want to talk a little bit about the data that we're going to actually assume that we start with. And the way that we extract this data is with convolutional neural networks. So our data is going to be a collection of positions. At every single frame of the video, you'll look at that image, point to all of the things that are in the image and locate them, okay? I'll show you a picture to see. Random number of objects in every frame. These indexes are arbitrary. Index 1 at time 5 has no relationship to index 1 at time 6. Right? We cannot follow an object by simply looking at index 1. There's no relationship. In fact, that's the whole problem of object tracking, is constructing that relationship. So back when convolutional neural networks were still sort of new, and I constructed one to locate objects, particles in a video, and I can use this neural network to track the nuclei. But diatoms are a little bit more difficult. So Anton Phipps basically constructed a U-net, right? That's what my paper here. I just used a U-net and applied it to something very specific in biology, right? I would never get this published today. It's funny, machine learning, everything moves so fast. But in 2018, this was exciting for those of us in math biology. Okay, so he was able to train this, and you get a bounding box, okay, for every object. And the bounding box is what? It's 4 numbers, right? It's the location and the size of the bounding box. So this is Z, right? And I'll use that notation, Z, throughout this whole talk for my measurements. Okay. In theory, Z could be anything that's observable with only one time of the video. Not velocity, but it could be orientation, shape, right? There's some things you could get from the bounding box. But for the most part, just think of it as being the location, okay?
So why is it unordered? I don't get it. You have a video, so you know the time trajectory. So why is the time points are unordered?
The time points are ordered.
Ordered, yeah.
This is one single time, right? So the ordering of the bounding boxes in here is arbitrary. But of course we know time. So now I want to move into multi-object tracking. We'll just assume we've got a good convolutional neural network that's giving us very few false positives, but probably quite a few false negatives in some circumstances. Okay. So what I want to propose, and I'm definitely not the first to propose it. This idea goes back to 1979. I don't want a single set of tracks. Like I'll call a track set, right? All the tracks that explain one video, right? I don't want just one set of tracks. I want a distribution of tracks, each one ranked by how likely it is, right? And then I'm going to use that distribution to estimate whatever I'm interested in, right? The lineages of dividing nuclei, the speed or the reversal rate of diatoms, right? Whatever scientific problem I have. Okay, because much of tracking is obsessed with getting one set of tracks that's the truth, right? I know I'm going to make mistakes, so I want to know what the uncertainty is like in diatom velocity with respect to the uncertainty in my tracking. What kind of mistakes am I making? Right? Are there certain situations or places where there's more uncertainty than others? I want to be able to keep track of that. So that's what a distribution can do for me. And this has been called multiple hypothesis tracking in the literature. And these ideas have been around a long time. The hard part of multiple object tracking is called the data association problem. And it's pretty simple. I'm going to come back to this later and describe it in more detail, but basically we need to be able to assign the measurements that we have to a set of hypothetical objects. I don't know, the number of objects is random, and whether or not an object gets a measurement, right? Because we have missing observations, so that's random. Maybe a measurement is a false positive. It doesn't get a sign. That's random. And so there's a lot of things that need to go in this, but simply said, right, each hypothetical object would get at most one measurement at every time. Right? And a measurement can only go to at most one object. Right? So those are some of the constraints. But we swept up the really hard part of multiple object tracking into one problem and gave it a name. Right? So there's a whole literature on the data association problem. One of the major branches of this is the joint probabilistic data association. And this basically, you gain computational tractability by sacrificing the full distribution. You marginalize over the many hypotheses. Right? So it's, you're compromising in order to actually be able to compute something. So I'm not going to do this. I want the full distribution. Right? That's my goal. I like to have a movie once every 10 slides. So I just throw one in there. Just, I don't know, just to keep things interesting. I did have a point there, but I don't remember what it is. So I'll move on. Okay. So there's a literature on multiple object tracking that arises from aviation. Right? So you're measuring objects with radar, and you're trying to keep track of multiple objects that way. And it's interesting when this literature goes back, all the really good stuff is in this aviation literature, okay? But the algorithms are really shaped by that application. And so you kind of have to see where it doesn't quite fit when you move it to a slightly different application. So here's, of course, airplanes have transponders, right? So, tracking in aviation is only important, I guess, for, I don't know, military. Missiles, right? You're in a jet fighter, you want to know where the missiles are, I don't know. So that application, you only care about what's happening right now, right? And what information do you have about what's happening right now, what's happened in the past? Right? So then this is like a filtering problem. You're trying to get the distribution for where the missiles are right now based on where things were in the past. Right? But we have a whole video. We're not doing real-time object tracking as the microscope is taking the video. No. The video gets taken, saved to a hard drive, and then sent to me, and then I can spend 2 weeks trying to track if I want to. Hopefully my method doesn't take 2 weeks to compute, right? But, right, so does that make sense? I have both the past and the future that I want to take advantage of when I'm doing tracking. And so the algorithms have to change in order to take advantage of future information as well as past. That's a major constraint. There are some other little quirks too. It seems like the false positive rate for radar information is super high. So that it sort of influences how things are working anyway. Does anybody know where this comes from? So Breaking Bad, any fans of the show Breaking Bad? It's like the famous scene where the airplane crashes, and he's the what do they call them? Traffic controller. Anyway, okay. So here's I want to get into the Bayesian stuff. Everything that I compute, I'm going to think of as a distribution, and this is the way I'm going to visually present a distribution to you guys, but it's also the way we compute things. So I'm going to use Markov chain Monte Carlo. I don't have a mathematical function that describes my distribution, but I can sample from it. So I'm going to use a set of samples, right? So here's a set of samples that's sort of modeling a Gaussian. And you can make this very concrete through an empirical distribution, right? So visually, I'm going to show distributions for the position of things I'm tracking as a bunch of sample points. And that's going to be confusing at first, but we'll get past it, okay? There are some good models for multi-object, Bayesian models for multi-object tracking. All three are good. Actually, I don't know what the pros and cons are. I'm still trying to get one to work, right? Still trying to iron out all the details for one of them. I've arbitrarily chosen this middle one to work with, but I am not making any claims about one being any better than the others. I would, for future work, to implement these different ideas and see their different strengths and weaknesses. But basically, the data association problem is essentially the same in all of those models. It's still the hard part. Okay, single track, easy problem. This goes way back. Wiener, the famous Kalman filter, right? This is a solution for a state space model, linear state space model. And there, this would be an example of a linear state space model, where these are both normal, and I think that these means have to be linear functions, right? And of course, these are trivially linear. I mean, this could be a little more complicated, but this is just an example. What I really want to show you guys here is how I'm plotting things. Okay, so this is how I'm going to represent the distribution for the position of a tracked object. Are these many tracked objects? No, this is one tracked object, one object. Many dots are samples of its position. So this isn't a bunch of objects, this is a distribution of the position. Okay? What's the plus symbol? Those are the measurements. The great thing about a state space model is I have statistically defined a distribution for the position at any time. In between two observations, before the movie starts, after the movie ends, I can sample the position at any time. That's pretty cool. So what happens here? When the plus sign appears, I've got a measurement, and the points start to contract, because when I've measured, that's when I have the least uncertainty. So the variance is minimal. And then it expands again, right, until it's about halfway in between the two measurements. And then it starts to contract before the next measurement happens. It's anticipating the future. Right? So you get this contraction, expansion. It's a distribution for the position. Does that make sense? One object. Okay. Now, so that was a single object problem. Here's the multi-object problem. I've already introduced this notation, right? This is the measured positions. I need to decide which of these are false positives and which are true positives. Because I'm going to ignore the false positives, but I need a binary random variable that tells me that, right? These are my hypothetical objects. Right, so for the moment, I'm going to assume that there's a fixed number of objects through the whole time series. okay? So it doesn't matter if an object leaves the field of view, I just stop assigning it measurements if it leaves the field of view. Right? So it's a very natural framework that handles that problem. I could track something after it leaves and then re-enters the field of view, right? So I stop assigning it measurements, stop, and then maybe there's a measurement after I'm going to show you an example of this later. So I have another binary random variable for every time for whether or not each of these objects is going to get assigned a measurement. Okay. And then here's the data association problem. I need a mapping from the observed objects to the true positive measurements. Okay. I want a distribution, and this is the sort of form the distribution is going to take, like a mixture. These weights are super important. Remember, I told you I want a whole bunch of hypothetical track sets, each one ranked by how likely it is. Well, this is how likely it is, the weight, right? In theory, this sum over n should be all possible tracks, right? However, this is a vast space. We can never hope to consider every possible track set. So I grew up in a rural area. There were lots of hay fields, okay? This is not, I doubt this is where I grew up, but I mean, I just got this from Google Images, but this looks like where I grew up. Think of this as a hayfield that goes on effectively forever. I mean, it's technically finite. So what I want are the best tracks, a little haystack of the best tracks. So here I'm kind of thinking of each little hay. You call it a hay, an individual, I don't know. Each one is like a track. So I want the ones with the highest weight. And that's going to be an approximation of this distribution, okay? So that's the computational goal, get the best tracks, okay? But the problem is the methods are imperfect. This is the plague of Markov chain Monte Carlo across the board. Regardless of what you sequential Monte Carlo, whatever, you get one of these things that has all the weight, right? One of these numbers is exponentially larger than all the others, right? So a distribution of one thing is not really a distribution, right? And that's not the truth is not that there's only one thing that's highly probable. There could be other very likely track sets that we just our algorithm isn't finding. It's getting stuck, okay? And I'll explain that in a little bit more detail and show you some examples. Videos. Okay, so this is the dividing nuclei, right, in the fungus cell. This is the most probable track set. So I'm not showing you a distribution here, but I just want to show you how I'm visually representing multiple objects. This looks like a little amorphous cloud, but it's actually a whole bunch of little dots. Are they all objects I'm tracking? No. Each color is a bunch of sampled positions of its position. So this is the distribution for the position of each of these objects. Okay. Why do they fly out? Because there's a missing observation. So then the uncertainty in the position increases. The variance increases when there's a missing observation. The plus symbols are measured positions. When you have the plus symbol, you have very tight distribution. Why do I have multiple red things here? The same object? It's actually surprisingly hard to make neighboring objects different colors. This is a plotting problem, okay? They're different objects. It's just hard to. Does that make sense what I'm trying to say? Okay. There's lots of collisions here, like here you go. There's a crossing, right? A whole bunch of crossings there, crossing there.
Do they destroy each other?
No. These are like water balloons, right? Moving around in a viscous fluid. When they bump up against each other, they don't explode or break. They just sort of they move past each other like two sumo wrestlers, right?
So the colorful dots are given by your algorithm, and the white dots are given by the experimental video.
Exactly. And the color is my extremely imperfect graphing, right? But one color is supposed to correspond to one object.
Can I ask one question?
Yeah, of course.
Color, how many dots did you use for?
Oh, good question. I think it's 50. It's like 25 or 50.
That is, if we use some high number for that.
More samples, the more accurate your distribution gets, but more computationally expensive. Was there a question out here? No? Okay. Okay. So this is just one track set. So I want to show you some results that we would only be able to obtain by looking at the distribution. So here, what I've done is I've identified an object as and given it like an ID, and think of the ID as like a color here at the beginning of the video. So it says, given where this object starts, where does it end up by the end of the video? So by the end of the video, you'll see colors ending up clustered around multiple nuclei. So that says the distribution for the position of that nuclei is very spread out, but now it's almost like a mixture, right? There's like these little clusters of where it could be. So a lot of uncertainty here in both of these videos are little clips of the previous video. One is up here and one is down here. And basically what I did is I just looked for a lot of uncertainty. And that's where it ended up. Why? The source of uncertainty here is pretty clear. This is the edge of the video. The hypha keeps going in reality, but we only image it, right, up to that edge. Right? And this is the growing tip of the hypha, we call it. One branch hypha, that's Greek, I think. So there's uncertainty here because of it's the growing end of the hypha. Does that make sense? So now we've identified areas of the video where there's a lot of tracking uncertainty. And that already by itself is extremely valuable, right? So the biologist might say, well, just ignore those regions then. But that might not be interesting. We may be really interested in what happens, what's different about the growing tip compared to the rest. So then we would at least want to know what the uncertainty in what we're estimating, what's the contribution from tracking error in that uncertainty, right? So that's another important question we might ask. Here's another example from the diatoms. Again, this is the most probable track set. And you can see that there are a lot more objects here. So we had to come up with some strategies and modifications to our algorithm so that everything scales really well with the size of the tracking problem, right? I wanted to scale essentially linearly with the number of objects that I'm tracking. Some of these Markov chain methods that have been proposed do not scale well. So I'll talk a little bit about how we do that. Because like the objects down here, they probably have absolutely nothing to do with the objects up here. They're totally independent. But you can't decide that ahead of time because these things move all around, right? So you have to compute everything at the same time. You can't just break this up. into chunks. Because the edges create a lot of uncertainty. Okay, so another thing to look at is that very often we can correctly keep track of things after they leave the field of view, and then sometimes they re-enter. Sometimes that's not correct, and sometimes it is. But again, this is just the most probable track set that we computed, right? Not that exists. We don't know that it's like the most probable track set overall.
So what's the little blobs behind the that?
Good question. I always have to explain this, everyone, and I always forget. Okay, these are diatoms. You kind of notice they move the same way. It's almost like they're casting shadows, but then it doesn't correspond to anything, right? Okay, so what this is, there's actually a thin layer of water over the auger, and diatoms will swim or they crawl. Okay, so they'll either swim at the air-water interface or they'll crawl at the auger-water interface. One or the other. They don't swim in the middle. So they're swimming in two layers, and it's so thin, this layer of water, that the diatoms on the other layer are just barely out of focus. And I'm pretty sure he must have chosen the layer of water to be just thick enough that these are barely minimally out of focus, that they don't interfere with the tracking. But I think that there is a spot somewhere in this video where you see, right there, there's a false positive, right? So you got to be able to ignore the false positives that get on these shadows when they do occur. So in aviation, this would be called clutter. I don't know what clutter, I mean, I know what clutter in my living room is, right? But clutter for a radar dish, I don't know, like rocks on the ground or something, maybe they generate false detections. It seems like those should be stationary and you would be able to point them out, but all right, that's what they call them anyway. So it's kind of funny that we get like clutter. But you always have false positives because in biology, the images, the videos, they're always going to have weird background like this. Like, how could you ever predict this if you weren't trying to solve a specific scientific problem? But something like it would be nothing like this in another application. But there's always going to be something, right? So you need a real scientific problem to see these problems. Oh, let me go back real quick. There it goes. It took a minute. So one of my favorite examples is this one here. So there's going to be a three-way collision here. And you notice that it's correctly resolved here. So all three of these objects are moving at different speeds. They collide at the same time, and they all switch. Right? That looks easy to track when we use our eyes. Surprisingly difficult to correctly resolve this with automatic tracking method. So again, I'm going to look at the distribution. That three-way collision is going to be here, okay? So I picked a model that actually accentuates the uncertainty a little, quite a bit. So the distribution is, for dramatic purposes, the uncertainty is increased. But the idea is the same as before. Given where an object starts, where does it end up? So you can see that by the end of this video, we're not sure where things are. So if we use a bad model, then we have a lot of uncertainty in this situation. It can't tell the difference between these different tracking scenarios. But at least with this method, with a bad model, it tells us it's a bad model, right? And if we have a good model, maybe there's only one really probable resolution to this. In this one, it's the edge of the video, and there's a lot of these that like leave and then re-enter, and so that's really and they collide, so that's the source of the uncertainty there. It's like I picked these two problems. I swear these cells in both of these problems are deliberately trying to evade tracking. Okay, so now what I want to do is talk about the algorithms and methods that we use here, okay? I want to reiterate that the main problem is that one track set gets all the weight in the distribution. That's the main problem with the practical problem with these Markov chain Monte Carlo. Of course, the truth is that there could be many track sets that are close to each other in probability, but we can't search them out. When we have an imperfect searching algorithm that finds a track set which is much more probable than any of the other ones that we looked at, right? And to put it simply, our sampling method gets stuck. That's what happens. So we have to find a way to get it unstuck. So that's one of the main problems. So I want to sort of describe on a very high level how we do this, okay? So this is what most existing good tracking methods use, okay? Something called, they're just finding the most probable track set, okay? But they don't even really do that, right? So we need to be able to leverage this and to be able to get a distribution out of it, and then to actually look for globally optimal, or globally sampled track sets. But this is such a good approach that basically every method tries to leverage this somehow. So this is linear programming. It's called the this is called the linear assignment problem, bipartite graph matching problem. It's known by many names. So here we're only looking at one time, and the idea is to match a set of objects with another set of objects. It's a really useful algorithm for lots of different things in microscopy. And there's a cost of assigning Zj to Xi, okay? And it's linear because this cost only depends on one pair. And of course, there's a cost of not assigning an Xi a measurement and not assigning a measurement to any object, right? So we have some parameters, and this comes from our statistical model. Right, so given this optimization problem, there's a really fast and efficient algorithm for solving it, called the one that is most used and available is one from Jonker and Mulganet, and it's just basically a variation on Dijkstra's algorithm, and it has a nice version of that can work with sparse data structures. So that's one we use. And you'll notice that this is a linear programming problem, and so this is the variable we're trying to optimize over, right? And we're not making any assumptions about this being integer values, right? So the idea is that this variable would be 0 if there's no assignment and 1 if there is an assignment. So these have to be either 0 or 1. A 0.5 assignment makes no sense. But we're able to use a real valued optimization problem, and we know that our optimal solution will be integer valued. It'll be either 0 or 1. And that's because this problem has special structure, right? And this totally unimodular, I guess is what they call it, right? So that's nice. We don't have to do what's called integer programming, which is a little more complicated. Okay. How do we turn an optimal assignment into a distribution? Well, there's a nice extension of this algorithm called the Murdy algorithm, and it's very similar to like what they call a branch and bound algorithm for doing integer programming, where you can get the k best. K is just any number, so I can find the k suboptimal solutions, right? And then that's not the full distribution, but you can stop it. You can get as many of them as you want until, the probability becomes so low that it doesn't matter. So you can truncate that distribution where you want. So this is a pretty effective way to get a distribution out of it. And then this algorithm just uses Jonker-Volgenet, right? It just leverages that to get the k-best assignments. So that's how we get a distribution. And then if we're sampling, we can just sample from that distribution. Okay. If they were that easy, then this whole thing would be solved, right? But that only works to assign objects at one time, right? So then we could do that for every single time. That's a greedy algorithm. Optimal, but it's only optimal at that one specific time. Okay, does that make sense? It's not globally optimal. Nevertheless, all good existing tracking algorithms do some version of this, okay? All right, so what's the problem with the nonlinear case? If we look over all times, now we add this time index to our variable that we're optimizing. The objective function now depends on all of the assignments over all the different times. So now it's nonlinear. So it's a much harder problem to solve. We don't have a linear objective function. So how do we do it? Well, our strategy, since what we want to do is sample anyway, we're not going to solve the optimization problem. We're just going to try and sample. So we use a Gibbs sampler. Okay. So then we sample the assignments for a fixed time, because that's linear, with everything else being fixed. Then we move to the next time and the next time and the next time. But we don't stop when we get to the end. We start over again and sample and sample and sample. And so moving through the entire time series is one step of a Markov chain. So then we take many steps, and so you can show that these will then converge to samples from the stationary distribution we want, right? We're not finding the optimum, but we are sampling, and that's what we want to do anyway. So does this work? No, of course not. Major problems. Like in theory, it works. But in theory, like if I had enough compute power and time that I could compute forever, on time scales that we can actually, do something, this is what happens. Here's the index for this track set. I've looked at 5,000 track sets. One is probability 1. All the rest, I didn't just manufacture this. This is actually the result of a tracking run. So this is not a distribution. I mean, technically it is a distribution, but it's not an interesting distribution because only one thing has any probability. And that's not because this is the truth, that it's truly the most probable track set. It's just the most probable one that I found. What happened? This Markov chain gets stuck. So it's an ergodic Markov process. In theory, it can go anywhere. Practically, there's like a region where it takes an exponentially large, in some sense, time to get out of it. In physics, we have this problem of diffusion in a potential well. And you want to wait until diffusion can cause you to exit a potential well. Like the hitting time of an Ornstein-Uhlenbeck process. Okay? These are all related concepts. That's what's happening here. This Markov chain is getting stuck, and so what we need to do is jump to more interesting areas of the phase space, right? And then start the process over again. So this is what we came up with. It's called a switch sampler. And this is just by observing some of the mistakes that get made. There's often just one spot where objects cross where the tracks make a mistake. So here it's just my little cartoon. Blue is there's a mistake here, right? So blue, 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 and but here, right? Blue is now attached to red. So if we could just switch it right here, and I think of this as like recombination in DNA, right? I just need to like switch everything in that spot, and then everything would be fine. But maybe there's another spot too. So I need to be able to keep sampling these switches, right? And this is, I'm thinking of this as like jumping to more interesting areas of the phase space for the Markov chain. So we have a little sampler that compares different tracks and allows swapping to occur where things are close. It's not based on distance, it's really based on the posterior, but we also formulated a representation of the track distribution that scales really well with the size of the problem. And this was inspired by column generation. And the idea is to keep a large set of potential tracks and then combine them together into potential feasible track sets using a linear programming problem, an integer programming problem. But I think I'm running a little behind, so I won't get into too much more detail about that. Okay, but here's a result. Here's my old problem. I already showed you this. And this is with the same model in both of these, but this is bad computation. This is with the switch sampler. But it's a fancy model. We use sequential Monte Carlo. And now, look, we have around 100 track sets that have significant probability, right? So that's much nicer. But what can happen? On the other side, this is something else that can happen. Here's my fancy model, same as the previous slide, this one here. But suppose I wonder, well, do I need my expensive fancy model? Maybe drift diffusion works much faster. I'm very interested in making that work because I don't need to run it for 5 hours on a GPU. 5,000 track sets, 1,000, 5,000. 5,000 track sets, all feasible, all with significant probability. Is that good? Is that better than this? That's terrible. It says that this model is so terrible, they cannot tell the difference between 5,000 different track sets. It's a bad model. It can't track. It can't, does that make sense? So I thought it was interesting that now that this thing sort of works, we can start to look at models and see which ones are good and which ones are bad in some sense. Got to be a movie to end it off with. I know this is not a good movie, and I'm sorry. I'm trying to add births, so now there's not a constant number of hypothetical objects. Right? We actually model the chemical reaction. It's a zeroth order. No, it's a first order reaction. And every time an object is born, there's like a light blue. I know it's a terrible video. I'm sorry. I'm working on it. But you can see when objects are born. So by the end of this video, the number of objects that we track, it increases substantially. Okay, to summarize, we can use velocity to resolve crossings and uncrossings. We can handle nonlinear models with sequential Monte Carlo. Seems to work pretty well in GPU. We can sample the full track posterior using Hamiltonian Monte Carlo. What else? We can even have hidden states using expectation max. No, this is for model parameters. That's right. If we just want a point estimate for our model parameters, we can do that with expectation maximization. And we can also, I didn't point this out, but we have the flexibility not just to model the motion of our objects, but the experiment itself. the field of view. There's a lot of different details about the microscopy that we can actually model that and get a more effective tracking algorithm. We want to do lineages to be able to resolve the cell cycle phase of a bunch of coupled oscillators. We can validate that with hand-labeled tracks. We want to be able to make some strides on handling nonlinear models in terms of making it more computationally efficient. And I think that this is like a really good avenue to look at. So banded Markov chain Monte Carlo, this seems to be taking prominence now in a lot of the methods I'm seeing out there. So I want to explore that. Anyway, if anybody has any advice, I would be happy to hear it. And sorry to end a little bit late.
Thank you. Yes, thank you for the nice talk. I would like to see again the old model and because of that something. Yeah, can I see the next slide? Yes, I have one question is when you see the video, do you have to choose the maximum index for the track?
Oh,
Can you?
yeah. So when I showed you the most probable track set in those videos,
Okay.
or before I would choose this one, right? Track index is totally arbitrary. I've just sorted them by how likely they are. I don't know if is that what you're asking?
Even if you fix the same video, the left side, the maximum number of track set index is 10,000, but the right side is 5,000.
Oh, totally arbitrary. I can look at any number. So I picked 1,000 because I could have picked 400. Right? I truncated it just because it's basically 0.
Okay.
This one, 5,000 because there's still significant probability there.
I see.
So I didn't go out far enough, but this took too long already. I couldn't keep going because the model's too terrible. It can't tell the difference.
Thank you.
Good question.
Any other questions? So your methodology purely based on data, not any diffusion or Brownian motion based model?
No, we model the motion. That goes into the tracking method. To get velocity, we have some model for how it moves, like a persistent random walk. They're basically switching what do we call these? They're like Gaussian mixture models, right? So then there's some hidden state that says switch direction, like in the case of the diatoms. There's like a 0, 1 hidden state. When a 0 moves forward, and then it switches to 1 and it moves backward. And something just slightly fancier for this, because this is tracking the nuclei, right? So it turns out there's basically 3 states of the nuclei. Either it moves forward, backward, or just does Brownian motion.
And what do you mean by here, simple grid diffusion model?
Right, so if you look at the nuclei, the tips grow, like I showed in the videos, and so then there's like fluid flow that goes at the speed of the growing tip.
Yeah.
And so the nuclei just sort of float along with that speed, but otherwise they do Brownian motion. Okay, but that's not the full truth because they also do molecular motor transport. So they also move forward and backward.
Yeah.
But I was curious how if I don't model the fancy stuff, the switching velocity,
Okay.
right? But I still get the fluid flow in the background. Because this is much faster. Is it good? And the answer is no, it's terrible. But you can start to ask like, okay, how do I make it better? Maybe the drift is a function of time, or right, you can try and make it better. But you make your model nonlinear, and there's a huge jump in the computation time. Right? So this has the advantage of being a linear model. So then you know the distribution analytically. It's like everything's Gaussian. Yeah, I'm sorry. A lot of these details I just didn't have time to put in the talk.
Another question? So I have another question, actually. So if you use a lot of samples for Monte Carlo, the variance becomes smaller. So when you visualize your video with the elaborate dots, why don't you use more dots to handle the uncertainty issue more clearly? For example, you mentioned in the middle of the talk, you mentioned that you use the 20 dots for each distribution.
My choice of the number of dots here is purely for visual purposes, right? I wanted enough dots that it was a good visual representation, but not so many that it was too cluttered. Yeah, but you're right. More samples, the better in general. So what we chose to plot is not necessarily what we chose to compute.
I have one question more is that I would like to in this talk I understand that mathematical model to track some behavior, but I would like to know why they want to track that their nuclei on that things, because I just wonder maybe they want to measure some they want to see some super diffusion or self diffusion of nuclei. Do they want to know that kinds of thing after tracking them?
Not with the nuclei. That is a question that I've asked in other situations. Okay, so the scientific questions, I think that's what you're asking. What sort of scientific questions do we have? So remember, in this case, the diatoms, we don't understand how they move. So we're just really trying to quantify and describe the statistics of their motion here. That will help us model and figure out how they move. So I'm interested in like, what is the velocity? So you can model the turning, how they turn as like a diffusion in the angle. It's a persistent random walk. So what's that diffusion? What's the frequency of reversal, right? Is there anything strange that happens when they collide? Maybe we can figure that out. What else? I think that's basically all for these guys. And then you know, the scientific questions here. Remember, we want to keep track of their lineage through time. That's our main goal, right? So we want to know where in their cell. So how long has it been since they divided? That's where they are in their cell cycle, right? And just to keep track of the lineages over time.
Thank you.
But we are also interested in like, there's background fluid flow. What is that fluid flow? Does it change in time? What happens when these cross, right? And we have a student who says that when two cross, what did he tell me? They tend to reverse velocity when they uncross. I don't know. Yeah, there's something weird about what happens when they hit each other. They don't explode, but they do something. And like how? So it might not be obvious, but they switch between different active transport, molecular motor motion states. We'd also like to know how frequently that happens.
Thank you.
You think this life of the fungus?
The life? Are you asking how long it is, or? Well, much longer than this video, hopefully. So it's really hard to do, actually. When you image, you are shining a super bright laser, and they don't like that. It kills them. If you shine too much of that laser, it kills them. So you've got to image enough that you're resolving this in time that you can keep track of all these objects, but not so much that you kill the fungus. So Grace is the one who, you know, optimizes really all of these constraints to try to get an effective video. So I don't know what the lifetime of these cells is. They live in tobacco plants. I don't know how long they live. Yeah, it's a good question. The cell cycle is like an hour. The,
Do they help with growing speed you are trying to?
the,
That speed is related to the growth of the.
oh, yeah. So how fast this tip grows is how fast they're growing,
Yeah.
right? And that determines fluid flow in this whole space. Yes, so that's true. That's a good point. Scientifically, that's interesting, right? The drift tells us how fast they're growing. And in a real organism, there would be different amounts of food in different directions. More food, more growth.
In your video, there is one case tip like to food.
This is the best video to see tips. It's not very bright though. Can you guys see it?
I can see. Yeah.
So as this video goes, you'll see these divide. These tips split into two. Is that what you mean?
Yeah.
I can see.
Can I what? Oh, yeah. I see what you mean. This one. Oh yeah. We track this as it splits. It splits I don't know, down here somewhere. So how do we track it when they split? We have to reconstruct this line, and then we create like a graph structure. So then we kind of glue together two lines. So by hand we identify where the tip split, and then there's like a pre-processing step. We get the geometry of the cell. That actually all goes into the model because we need to model the fluid flow, but the fluid flow is linear inside of the hypha, so we need to know what that is. So we have to account for all of that. Yeah, it's a lot easier when they don't split. But I mean, there's lots of questions we have about what happens when they do split. Remember, they try to keep a constant density. That means that the density has to like temporarily double, right? And then they split apart.
Yeah.
So sorry, we've gone way over time.
Just formal Q&A time. But is there any additional questions? Okay, if not, let's give a big thank to the speaker.
Thank you, guys.
What? We usually work on this project